wanted to kind of check your slides, they're already loaded up there if you wanted to get up there. Another thing I'll suggest you do too is just kind of see where the podium is placed. If you're okay doing it from the podium and kind of, being that far away from the slides, that's fine with me. If you want to take the handheld and kind of do it from over there, that could work for you too as well.
Okay.
Are you looking at speakers?
Yeah, we're both.
I am Mike. Yeah, I'm emailing you incessantly.
Oh.
Ritisha. Great.
Well,
Yeah, so let me just double check. So Domonique, Olivia,
Valle.
Valle, got it. Okay, and Ritisha Day, is that correct? Okay, good. All right, well, thanks for coming.
Kendra.
Kendra.
Yeah.
Excellent.
Nice to
Great.
meet you.
So, if you guys wanted to go, you can check your slides. They're all
Oh, yeah.
they're all loaded up there. Also, see where you think you would prefer doing the presentation. You can do it from the podium. You can also do it via handheld mic. So it depends on what is most comfortable. Step on up, just take a look around.
I'm Grant. Nice to meet you. Do you guys have clicker?
We do have a clip.
Awesome.
So you kind of have to, have two hands going if you have the handheld mic, but
Sure.
but it should work out.
Do you have two mics?
So there's a mic at the podium, but then we just had to have one mic. Yeah. Oh, that's a good point. So you will have to do handoffs, I guess.
Okay.
Yeah.
Okay.
In case I need help with anything, so I wasn't stressful. It wasn't just physical chemistry test that I have to do by midnight tonight. Took me a minute to figure out why. Yeah, I'm missing biochem.
Kendra, your last name?
Wyant.
Wyant?
Yeah.
What's your year? What's your major?
Chemistry, ACS emphasis.
Nice.
What about you?
I'm a biochemistry, molecular biology.
I envy that.
Just so you know, so if you want to, you can go up and check your slides. I would suggest you go up and see the podium feels to you.
Yep.
It's a little farther from the screen, so you can decide to either do it from the podium or you can do it via handheld mic.
This would be my
There's a
fault.
power switch on the side of it, but I may have got turned off, so.
I see. Okay, cool. Cool. Are they just kind of sequential in the same slide deck?
They are, yeah. So, yeah, depending on where you fall, you'll have to kind of go through everybody's slides.
And I've done one standing presentation.
Cool. Looks good. Anyone else want to go through it?
I'll use the clickers on.
Yeah, test it around.
If you need any water, there's water up there for you. There's also some more outside too. our first time doing it in this venue, so there's some lumps. But yeah, but I think if you take the handheld mic, it should be easy. I just have to remind everyone to use good mic technique. Make sure you kind of keep the mic close to your mouth when you move around. It's often hard for people to remember that when they're trying to remember other stuff, right?
On right now.
Undergrads go first. Undergrads.
Hello? Hello?
It's on.
I'm the one that cooked the orders, but I don't know why I did what I did. I had my reasons at the moment, but it's foreign to me right now. Yeah. Well, somebody's got to go last. That's you. Somebody's got to go first. All right.
How strict is this 30 minutes?
Yeah.
There's not going to be a timer. Nobody's going to pull you off stage.
This is a good 30-second
This is as relaxed as a presentation can be. All right? You have nothing to be concerned about. All right. So Matthew Dubin. Does anyone even know or see Matthew? This is Matthew. We're also missing Mindy. Matthew?
Yes.
All right, perfect. So if you want to go back through Emmanuella, check your slides. And then also I recommend you sort of stand at the podium, see how that feels, because we have two options for mic. So either you're at the podium or you grab the handheld and kind of stand up, stand wherever you want. Yeah, and we have a clicker and so.
Sounds good.
Yeah.
Thank you.
Okay, no problem. Okay, so for some reason it's sort of disconnected here. Maybe, I'll get some. Great. Definitely gonna be here later. The laptop seems to be on different slides.
All right. Yeah, there was multiple PowerPoints open. That was, yeah. So one was still in presenter mode while the other one was, so now there's one PowerPoint open. So there you go. Yeah.
I would recommend you definitely go to the podium. There's two mic capabilities, so you can talk into the podium mic. There's a kind of
Good morning, everyone. I'm honored to be here on behalf of Automated Logic, proud member of BioForward. At Automated Logic, we're deeply committed to supporting the biohealth industry right here in Madison and across Wisconsin, where innovation is shaping a healthier future. Today, we're thrilled to sponsor the 4th annual communications competition, which shines a spotlight on some of the most promising voices in healthcare and biohealth. These students are not only the next generation of researchers and innovators, but also crucial players in advancing the technologies that power this industry. Their dedication and fresh perspectives are a key in keeping our biohealth sector strong and resilient and ensuring that Wisconsin remains a leader in medical and scientific progress. I'd like you to give their full support to these talented individuals as they share their research and inspire us. Thank you, and I'd like to introduce Mike Harrison from BioForward.
Hello, everyone. I'm Mike Harrison. I'm the head of the Milwaukee office of BioForward and the director of strategic initiatives and community outreach at BioForward. This is such a special session for us. We've been doing this as a 4th year, and it's really grown ever since we got it first started. You know, it's so special because it's really one of the few chances that BioForward really gets to highlight these not only the exceptional research being done here in the state, but also the scientifically minded students that will, eventually make up the backbone of both the research community and the biohealth industry. Like I said, it's grown since we got this started. I remember the first year we were just doing graduate students, and we had students from the Medical College of Wisconsin and students from UW-Madison. This year we have undergrads and graduate students, and they came from both of those institutions, as well as Marquette University, UW-Stout, UW-Whitewater, UW-Eau Claire, and UW-Milwaukee. So we're really seeing a much wider breadth of experiences and a much more diverse group of folks. So it's a great joy to go through all these videos. So there were many applicants this year. And what you're going to see today are 5 finalists in the undergraduate category and 5 finalists in the graduate student category. So these people are already winners. So they've all already won. They've already accomplished. We looked over their videos. The videos have been judged by a group of judges that are made up of biohealth leaders and biohealth board members. So we already know who's won, but we're here to present their work to you, the biohealth industry, so you can learn a little bit more about these fantastic people and the work that they're doing. So let me get things started there. So we'll start off with the undergraduates. The first undergraduate presentation is actually a unique one right off the bat. So it's our first co-presentation. So we actually have two kind researchers coming up here, Ritisha and Domonique Olivia Valle from the Department of Biological Sciences at UW-Milwaukee. Come on up.
Hello, my name is Ritisha.
And my name is Olivia, and we worked with the bacteria Xenorhabdus centyromate to identify novel antimicrobial compounds. Antimicrobial resistance, also known as AMR, is a global threat due to the historical overuse of antibiotics. Bacteria are becoming increasingly resistant, making it difficult to treat deadly infections. AMR is a growing problem. Millions have died due to antimicrobial resistance, and this number is predicted to surge to 10 million by 2050 if we do not address this pressing issue. That's why there's an urgent need to discover new antibiotics with new chemistry from new sources.
One potential new source of antibiotics is the bacteria Xenorhabdus centyromate.
Xenorhabdus centurme lives in the guts of a nematode. Nematodes infect insects, and Xenorhabdus is released into the blood of an insect, where it releases toxins and antimicrobials, which kill the insect. So we took the cell-free supernatant from Xenorhabdus centurme and spotted it on agar plates and overlaid bacteria on top. And then you can see clearance zones, which indicate the presence of antibiotics, since bacteria are unable to grow there. So we took the cell-free supernatant of X centurme and added amberlite beads. And what amberlite does is it binds to those antimicrobial compounds so we can later elute them with solutions. And then we were left with partially purified antimicrobial compounds. So we wanted to test whether these compounds were able to kill antibiotic-resistant bacteria, and we found that they could. They were able to kill methicillin-resistant Staphylococcus aureus, also known as MRSA, Klebsiella pneumonia, and pathogenic E. coli.
So our next goal was to identify the genes involved with the production of antimicrobial compounds. So using genomic analysis, through genomic analysis using bioinformatic tools, we identified 17 non-ribosomal peptide synthetase genes named as STI through STI, which are involved in the production of antimicrobial compounds. Then our subsequent RNA-seq analysis showed that the genes STI, STI2, STI10, and STI17 were all upregulated during the antibiotic-producing stage.
So in conclusion, we developed a novel protocol for antibiotic purification and also identified an antibiotic active against multiple antibiotic-resistant bacteria.
And we identified 17 biosynthetic genes for antimicrobial compounds, some of which are upregulated during the antibiotic-producing stage. Thank you for listening, and these are our mentors.
Thanks a lot. Okay. The next presentation is from Foster Boom, who's a part of the Department of Chemistry at UW-Whitewater.
Hello, everyone. So I'm going to start by passing out a few samples I have, because I know chemistry is a little bit of a hard thing to follow. And so I want to give you guys a good visualization of what our implants end up looking like. So start there. All right, so my name is Foster Boom. I am a chemistry student at the University of Wisconsin-Whitewater, and I conduct undergraduate research with Professor Stephen Gerard. My project is enhancing the bioactivity of titanium implants via silicide bioglass coatings. So osseointegrated implants are currently eclipsing the older socket implants. Osseointegration simply means the implant is directly inserted into the skeletal structure, and titanium is commonly used for these applications because of its robust mechanical properties, but it has low bioactivity, so it relies on coatings to improve osseointegration. The problem is the current approaches to coatings like polymers and bioceramics have some pretty significant downsides. So polymers have hydrophobicity, which reduces the strength of the implant site, and they also present potential toxicity concerns. And bioceramics are hard to coat onto complex geometries, and they do not regenerate new bone tissue. So bioactive glass is a substance that was developed in the 1960s that has an extremely porous structure that allows for the regeneration of new bone. However, it can't be coated onto metal using current technologies like plasma spraying and enameling. And it also is extremely easy to create crystalline bioglass and reduce its bioactivity. So our approach is to create an intermediate layer between the metal and bioglass by using molten salt electrochemical methods for the reduction of silica, to generate a titanium silicide layer. Why is this important? Well, when we generate a metal silicide in this way, we also generate a thin and robust native oxide layer that is grown and chemically bonded to the sample. This substrate can act as an ideal place to grow bioglass onto. So in theory, we should be able to develop a titanium implant that is chemically bonded from the titanium to the titanium silicide to the native oxide and to the bioglass. So when we conduct this experiment, we take our titanium silicide samples and we introduce them to our bioglass synthesis route. We can see that the bioglass does, in fact, chemically bond to our titanium silicide layer, and the reality is perfectly reflective of what we theorized. So this demonstrates a promising new coating for osseointegration of titanium implants. Just to make sure that our samples retain their bioactivity, we conduct experiments in simulated human body fluid according to our particular ISO. And when we do this, we can see the formation of bone crystals, synthetic bone crystals 1 to 5 microns in size, which indicate that this will very likely regenerate new bone in the human body. So this could impact millions of people through better osseo-integrated titanium implants. And it could also see a benefit in the field of magnetic resonance imaging due to the diamagnetic properties of our coating. If you'd like to talk more about that, I can talk after the presentation. So thank you for listening.
Okay, our next talk is Jadyn Hartwig from the Department of Psychology at UW-Eau.
Hello, everyone. My name is Jadyn Hartwig, and I'm a sophomore undergraduate studying at the University of Wisconsin-Eau Claire. I'm also part of the Wisconsin Addiction Research Laboratory, studying the effects of alcohol on aged rats. According to the CDC, alcohol is a major public health issue in the United States. Excessive alcohol use caused around 178,000 deaths each year, making it one of the 4 most preventable causes of death. According to the NIH, Wisconsin has a higher than average adult drinking rate of 64.4% compared to the national average of 55.1%. This ranks Wisconsin as the 3rd highest state for adult alcohol consumption. Economically, binge drinking in Wisconsin costs almost $4 billion every year. In 2022, 924 individuals from Wisconsin died from suicide. 1,459 died due to opioid overdoses. 3,331 individuals died from alcohol attributable deaths. Alcohol misuse is the epidemic we decided we could live with. The average age of Wisconsin and this country is increasing, and for the older population, the rate of alcohol consumption is not only expanding, but at a faster rate than younger age groups. According to the Wisconsin Department of Health Services, this current age population is growing and is projected to double from 2010 to 2040. It is also important to consider the fact that older individuals use healthcare significantly more than their younger counterparts. Link the increased size of the aging population and increased alcohol use within the current age population, resulting in a large-scale burden on the healthcare system, a critical public health issue. There is a vital need to understand the impact of alcohol, specifically looking at the aged population. Our lab, through a variety of standard lab testing methods, including models analyzing movement and cognition, can assess the impact of alcohol use. Our most recent research has shown a variety of different effects and the effects of age and sex differences. Most significantly, we have found a variety of different sex differences between aged male and female rats. Our most recent research has shown a variety of different effects and the effects of acute alcohol exposure on aged male and female rats. Stop for a second. Testing motor impairment, we have found a result of alcohol exposure in aged animals are more impaired than adults or adolescents. We have also found a significant sex difference in that aged male rats are significantly more impaired by the effects of alcohol than aged female rats. This specific sex difference is groundbreaking, and we are continuing to analyze it from a variety of different results. In another experiment analyzing non-spatial learning, we have found varied results of the effects of acute alcohol exposure, in that aged female rats and aged male rats have the same amount of effects. We are continuing this investigation on further sex differences and the possible biomechanisms at play. In addition, we have identified one peptide in the brain which has evidence of attributing to the sex difference. Overall, this research uncovers both the sex and age-dependent effects of acute alcohol exposure. This data can be applied not only in the healthcare field, but in the production of pharmaceuticals and in the application of alcohol care and treatment. My research is dedicated to providing the necessary information to the pharmaceutical, alcohol treatment, and scientific communities, getting the necessary data out to those that need it to help end this undiscussed epidemic. Thank you for listening. I would also like to thank my amazing lab group, mentor, and co-collaborators that made this research possible. My name is Jadyn Hartwig, and here are some sources.
Okay, our next presenter is Shaun Harrigton from the Department of Biochemistry at UW-Milwaukee.
Okay, hi everyone. My name is Shaun Harrigton, and I'm majoring in biochemistry. I work with the Arnold Group, and today I'm going to be discussing how we're currently using biochemistry to help in the aid in the treatment of asthma. Okay, so first, I think it's very important to remember how serious asthma actually is. Over 339 million people worldwide are affected by asthma, and the health complications that it can cause, lack of diagnosis and treatment can lead to health complications and even death. And I think it's also important to remember how crucial asthma is related to inflammation, because with inflammation, it's an essential body mechanism that, the body's immune response to injury or harmful stimuli characterized by swelling, redness, heat gain, which can become chronic, and that's basically what asthma is. So, in our research, our main focus is TSPO, and that is also known as the translocator protein. So we have TSPO in our brain and lungs, and that's what we want to target because it's directly related to inflammation. That means when TSPO levels increase, so does the inflammatory response. And we can target the TSPO protein by using something called a ligand. So, ligands are molecules that bind to proteins, and typically, when they bind to a protein, it results in a biological response or a signaling event. And with that, we want to create a ligand that selectively binds to TSPO in the lungs. However, we have TSPO in our brains and lungs, which means that we need to create a ligand that can't pass through the blood-brain barrier. And with that, it'll be able to target the TSPO in our lungs only, which is where the problem is currently happening. So our progress, my involvement in this research has been more in the beginning stages involving the organic chemistry. So far, we've identified the most efficient system for creating these new compounds or ligands, but we still have a long way to go. We've also used other aspects of biochemistry, such as liquid chromatography, NMR spectroscopy, and mass spectrometry to help us identify and purify these new compounds. And so far, we've synthesized and tested numerous compounds. We've had many failures and successes along the way, but as you can see from our percent yields that we've had low and high ones. But in the end, our goal is to create a new ligand that can selectively bind to TSPO in the lungs and hopefully reduce inflammation. And I believe that looking closer at biochemical processes in our body, we'll be able to develop further therapeutic methods to hopefully help patients suffering from these chronic lung diseases. And that is all I have for today. Thank you.
Again, our final presenter in the undergraduate group is Matthew Dubin, who's with the Department of Genetics and Genomics at UW-Madison.
Hello, my name is Matthew Dubin. I'm a junior at the University of Wisconsin-Madison, and I'm studying genetics and genomics. Today, I want to discuss the revolutionary use of AI deepthink in single-cell RNA genotyping. So, to explain what RNA is, RNA is a molecule that is very similar to DNA and contains much of the same information as DNA, but has a much wider variety of uses within the body. One of its primary functions is to transfer information from DNA to biological machinery, which is then able to build molecules which are called proteins. One of these proteins is called insulin. This is a hormone that regulates blood sugar and is commonly associated with diabetes. Diabetes is a disorder where the body doesn't produce enough insulin or produces an insufficient amount of it. Here, RNA is very important as mutations can happen which can cause RNA to be incapable of transferring the information properly, which can result in a reduction or a complete absence of insulin production within the body. With diabetes affecting 38.4 million people in the United States, according to the CDC, understanding how and why this happens is extremely important to public health. One way that we're able to understand what's happening with our RNA is through something called single-cell genotyping, or single-cell for short. RNA genotyping is a method of taking RNA from a tissue sample and reading the genetic data, giving us the ability to see what's happening with our genome and how it is processed. Additionally, using single-cell, we're able to tell apart different cell types, giving us a more specific understanding of how RNA is acting in our bodies. The problem with using single cell, though, is that it produces an almost incomprehensible amount of data. There we go. With the most cutting-edge genotyping being able to read from 10,000 to 50,000 genes per cell, and with thousands of cells in a sample data set, data sets can reach up to 1.5 billion data points, compared to the traditional method of genotyping that gives only 30 million data points. This is where AI comes in. Current use of AI in genetic research is focused on a subset called DeepThink. DeepThink uses a network of different algorithms and programs connected in a similar way to how neurons are connected in the human brain. The AI is able to understand this data from unique perspectives and can carry out many different statistical analyses that would not be possible for humans in the same time frame. Typically, it would take many years in the combined research of a plethora of different organizations and institutions to recognize the connection that exists between different genes. Now this can be done in a matter of days with increased scale and accuracy. I want to revisit the insulin example. If we could figure out the genetic reasoning why some are born with diabetes, this would be something that could help millions of people. DeepThink in this case would be able to take large data sets from many different samples and make detailed and large-scale connections between the RNA data, its specific cell type, and how it's expressed physically within the patient. The utilization of DeepThink will propel research forward at unprecedented rates and will make RNA genotyping not only more effective, but it can increase the speed of otherwise time-consuming and labor-intensive research. Single-cell genotyping has the ability to change the world for the better, increasing our understanding of debilitating diseases, how our body works, and our history as a species. With these new AI tools, the progress we can make is boundless. Thank you.
Okay, so that was the last of our undergraduates. I just want to briefly say, so this competition is about communication skills. So obviously there's some very exciting research being done, but we really wanted to focus on a very critical skill that all of these researchers and future workers in the biohealth industry would be using on a daily basis. And it's especially important, in this day and age that as scientists we are able to communicate what it is that we're doing, and most importantly, why it matters so much that we do it. So I'm very thankful that all these students are willing to share this with you today. So. Without any further ado, our first graduate student presentation is Mindy Kim from the Department of Physiology at the Medical College of Wisconsin.
Hello, everyone. My name is Mindy Kim, and I'm a graduate student in the lab of Dr. Z Zhang at the Medical College of Wisconsin. And today I will discuss the novel link between a fibrinolytic protein and atherosclerosis. Cardiovascular disease, or CVD, is the leading cause of death worldwide, and 4 out of the 5 CVD deaths are due to heart attacks or strokes. And one of the main causes of CVDs is atherosclerosis. Atherosclerosis is the buildup of fats and cholesterol in and on the artery walls. This buildup of lipids can then deposit within the vessel wall, leading to the formation of atherosclerotic lesions. When these lesions further progress and become more unstable, they can spontaneously rupture, leading to the formation of blood clots, blocking blood flow to organs such as the brain and the heart, for example, leading to what are known as strokes and heart attacks. The discovery of tissue-type plasminogen activator, or TPA, and the ability to utilize its synthetic form in the clinic revolutionized how we treat strokes and heart attacks. TPA is an important fibrinolytic protein in fibrinolysis, or the physiological process that breaks down blood clots. By administering TPA, we are able to efficiently break down blood clots and restore blood flow to the rest of our body. However, there are caveats to this treatment. TPA is primarily effective when administered within 4.5 hours of the first signs of a stroke, and there are bleeding risks. Therefore, it is crucial to continue pursuing research in this field. TPA was traditionally found to be derived from endothelial cells, or cells that line the inner wall of blood vessels, upon acute vascular injury. Intriguingly, our lab discovered that TPA can also be released by the liver and assist with fibrinolysis. But even more than that, our lab wanted to identify what exactly is the physiological function of TPA within the liver itself. Through research in our lab, we identified that liver-derived TPA can regulate how fats and cholesterol can be packaged and delivered from the liver to the rest of our body. And now we became curious. Can we take advantage of this newly discovered mechanism to serve as an alternative therapeutic approach for cardiovascular disease? And to summarize, increased levels of fats and lipids in the blood can lead to the formation of atherosclerotic lesions, and further progression can lead to blood clot formation. TPA first revolutionized how we treat heart attacks and strokes by efficiently breaking down blood clots, yet its utility is limited by a restricted time window and bleeding risks. Our lab identified that liver-derived TPA can regulate levels of fats and cholesterol in the blood, and now can we take advantage of this mechanism to create a treatment that is more preventative rather than reactive? Our lab is excited to continue pursuing research in this field to regulate levels of liver-derived TPA with the hopes of preventing atherosclerosis and the more severe consequences of heart attacks and strokes. Thank you.
Our next speaker in the graduate category is Kendra Wyant from the Department of Psychology at UW-Madison.
Oops, sorry, I went backwards. Okay. Hi, my name is Kendra Wyant, and I'm a graduate student working with Dr. John Curtin at the Addiction Research Center. In 2022, over 30 million US adults had an active alcohol use disorder. Only 1 in 20 received any treatment. Moreover, recovery is a nonlinear process that extends beyond initial symptom reduction. Relapse risk monitoring could help patients identify lifestyle, behavior, and support changes needed to fit their current needs. But we don't have the resources for long-term clinician support, and self-monitoring is really hard. Machine learning models for adaptive, continuous monitoring may be one potential solution. Imagine a smartphone app that could alert patients of changes in their relapse risk and make personalized recommendations based on the top features contributing to their risk, like an urge surfing recommendation for someone with strong cravings. We assessed how well we could predict alcohol relapse in the next week, day, and hour in a sample of 151 participants with moderate to severe alcohol use disorder. Participants completed 4 daily surveys for up to 3 months. They reported the greatest intensity of craving, arousal, and valence since their last survey. Any stressful situations, risky situations, or pleasant events since their last survey, and future stressful events, risky situations, and efficacy looking forward into the next week. They also reported any alcohol use. These first 9 items became our features. Time and day of alcohol use was used to generate our outcome labels of relapse in the next week, day, and hour. Our features were fed into XGBoost models to make predicted probabilities of relapse. And we did this all using a resampling method to ensure that our models were evaluated with new data from new individuals. Our models performed exceptionally well. Area under the ROC curve is a performance metric of accuracy. Values range from 0.5 to 1, with 0.5 indicating chance performance and 1 indicating perfect performance. Our models had areas under the ROC curve of 0.90 to 0.93. But we can also look at how much an individual feature is increasing or decreasing a probability prediction. And we can use the top features to inform targets for clinical intervention. So for example, for this person, they have experienced a recent stressful event and it is increasing their probability of relapse. For them, we might want to recommend a guided relaxation activity. However, for this person, they also have experienced a recent stressful event, but they also report high levels of craving and low arousal and mood. For them, we might want to increase the number of pleasurable activities they have going on. But now, for this same person, 2 weeks later, they now have had 2 recent relapses, and their reported efficacy in their ability to maintain abstinence has decreased. So now, motivational enhancement techniques to help them re-engage with their recovery goals may be more appropriate. In conclusion, we can predict probabilities of relapse at varying levels of temporal precision with excellent performance. And we can dynamically characterize the top features contributing to these risks, or these probabilities of risk. Finally, these results suggest the feasibility of using machine learning for long-term monitoring of relapse risk. Thank you.
Our next presenter is Grant Nickles from the Department of Medical Microbiology and Immunology at UW-Madison.
Thank you. So, how do fungi interact with the world? I want you to imagine you're a fungal cell floating around, consuming nutrients, when suddenly 2 scary bacteria are trying to mug you. What are you going to do? Are you going to grab a weapon? Well, too bad, because you don't have any hands. But you can secrete powerful chemicals to protect yourself. In fact, plants, bacteria, and fungi have been widely called the world's best chemists due to the large variety of compounds that they can synthesize. And in fact, over 50% of FDA-approved drugs are directly or indirectly derived from some sort of natural product, with 18% of those specifically coming from a fungal source. And to go back to UW-Madison, you may or may not know this, but professors at UW-Madison actually invented the ability to mass-produce penicillin, the world's most famous natural product that I'm sure all of you have consumed. And this discovery has actually been argued to have saved more lives than any other medicinal advancement in human history. Transitioning to the 21st century, we have a massive problem, and that is that we are having a really hard time finding new medicines. And I think this is best exemplified by looking at the rates of new antibiotic discovery over time. In the case of antibiotics, we haven't been able to locate a new class in the last 30 plus years. And the major reason for this is we keep rediscovering the same drug candidates. On top of this, when we are able to find a novel compound, it's increasingly common that it'll be really similar to something that we already know with just minor changes to functional groups. What we started to realize in the 2010s is that the vast majority of drug candidates that an organism can make in the wild are just never produced in a lab setting. In the case of fungi, they'll actually only produce about 10 to 20% of the drug candidates that they can make in a lab. So this is why if you look at modern natural product drug discovery papers, they'll often follow a pipeline like this. You'll get your organism of interest and sequence its genome. You'll then run its genome through some kind of AI model that's been trained to identify drug candidate metabolic pathways, after which you can use targeted genetic engineering to turn on these discovered pathways that are typically turned off in a lab setting. So transitioning to where my lab comes into the picture, in 2018, previous researchers in my lab actually found a new type of natural product pathway within fungal genomes. And this was new because it just simply was never turned on in conventional lab settings. However, the issue was that it was so novel, it had never been incorporated into any of the training data sets used to predict drug discovery pathways within genomes. Or worded a little bit more simply, it was just completely invisible to us. So this is where I come in. I spent 3 years basically developing my own genome mining algorithm, catered to find this new pathway within fungi, and ran it on every fungal genome I could get from the internet. It was about 4,000 genomes. And to my lab's surprise, this new class actually ended up being the 5th biggest type of natural products that fungi can make. This means that there is a massive untapped potential for us to find truly novel drugs with truly novel chemistries by going after these. And to give you just a teaser, one of the compounds that I've actually followed up on from my predictions is proving to be incredibly potent against MRSA. MRSA is one of the nastiest antibiotic-resistant bacterial pathogens we have in the country, hospitalizing over 300,000 Americans annually and costing the US over nearly $2 billion in estimated healthcare costs. Once again, my name is Nickles. I am graduating very soon, and I'm very much so looking for opportunities, so I gotta sell myself. Come find the guy with big hair if you think my skill set could interest you in any way. I'm open to postdocs or industry. Thank you.
Okay, and up next is Claire Radtke from the Department of Biological Sciences at Marquette University.
Since the day we are born, our bodies work hard to keep us healthy. So then, as we age, why are we more likely to get fatal diseases like Alzheimer's and Parkinson's? To address these age-related diseases, scientists need to understand how our bodies change as we age. And the answer is in our cells. Our cells rely on proteins, which are large molecules that hold very specific shapes, allowing them to do important jobs inside our cells. Think of a protein like a paper airplane. A paper airplane must be folded to fly, and when it loses its shape and unfolds, it can no longer do its job. The same is true with proteins. But as we age, our proteins are more likely to unfold, and then they can clump together, forming what scientists call protein aggregates, which can then disrupt cell function and cause diseases like Alzheimer's and Parkinson's. Thankfully, science has shown that our cells actually have a way of stopping this protein unfolding and aggregation. So here enters the star and the hero of the show, the molecular chaperones. Just like a parental chaperone or a babysitter, these chaperones make sure that your proteins are behaving. But as we age, this system weakens, leading to more protein aggregation. And while we know that chaperones are really important, it's not fully clear how they work. So understanding their function is critical to explain their decline with age and then find ways to restore them. So my research at Marquette University is exploring some key molecular chaperones in a largely understudied disease. Transthyretin amyloidosis, or ATTR, is similar to diseases like Alzheimer's and Parkinson's in that it's caused by protein aggregation. But it has symptoms ranging from heart complications to muscle weakness and is very understudied. So my research shows that some of these key molecular chaperones may target the ATTR protein aggregates. And then also I have data that shows that they may physically reshape the protein aggregates, implying that they could be used for therapeutics. So if these chaperones can be used to break these aggregates apart, then that suggests that they could be used to prevent or reverse the harmful effects of protein aggregates and ultimately improve patient health. So my research on chaperones and ATTR is just one piece of the puzzle. Together, scientists are investigating molecular chaperones and their role in many different diseases caused by protein aggregation. And these ongoing discoveries can offer hope for future treatments that could one day change lives. So with that, thank you for your time, and I'm at table 9 at lunch if anyone wants to chat. Thank you.
Okay, and our final speaker of the day is Emmanuella Dwomo Agyei from the Department of Cell Biology, Neurobiology, and Anatomy at the Medical College of Wisconsin.
So, do you know that in the United States, about 15,000 fetal deaths are recorded annually? Now, about 35% of these deaths are due to complications of pregnancy. While these complications contribute significantly to fetal mortality, most still remain poorly understood. So, that is where my lab comes in. One of our lab's research goals is to better understand pregnancy complications. Today, I want to talk about one of such complications that my research focuses on, and that is placental abruption. Placental abruption is the premature detachment of the placenta from the uterine wall while the baby is still in the womb. As you might already know, the placenta is an important organ of pregnancy that supports nutrient and waste exchange between the baby and the mother. So you can imagine, if the placenta separates before delivery, that can result in some severe consequences. It can lead to growth restriction of the baby, where the baby does not grow to its expected weight. The mother can bleed a lot, and unfortunately, it can lead to the death of both the mother and the baby. So you might already be wondering, or asking this question that I have been asking myself. Emmanuella, how does placental abruption occur? And the answer is, we still don't know. It is still a puzzle that needs to be solved. However, we do know of certain factors that increase your risk of developing placental abruption. One of such factors that has been long associated with abruption is a condition known as thrombophilia. Thrombophilia is when you have an increased tendency to form blood clots. Now, the association between thrombophilia and abruption has widely been shown in several human studies. However, in our lab, we have developed a genetically modified thrombophilia mouse model, and interestingly, these mice also exhibit placental abruption. Therefore, confirming that long suspected link between thrombophilia and placental abruption. So using this thrombophilia mouse model, my research aims to ask the question, how does thrombophilia lead to placental abruption? So far, we've studied the placentas of these mice, and we've observed that compared to the placenta of a healthy mouse, the placentas of our thrombophilia mouse have blood clots, not just blood clots. Within the blood clots, there are immune cells, specifically a type of immune cells known as neutrophils. And interestingly, this same observation can be made in human abruption cases. So, considering the potential role of neutrophils in promoting blood clots and also causing tissue damage, one of the key questions we are currently trying to answer is whether neutrophils are key contributors to placental abruption development. And to address this question, we plan to deplete or get rid of neutrophils in our thrombophilia mouse and see if that will prevent or stop placental abruption development. So in summary, my research aims to shed light on the mechanisms driving placental abruption. By understanding these processes, we hope to contribute to 1, a better diagnosis, 2, prevention, 3, treatment strategies that will ultimately improve the outcomes for both the mother and the baby. Thank you.
Okay, well, that concludes all the presentations, so there's only one thing left to do, which is announce the winners. You know, I did run into a couple of the judges here at the summit yesterday, and they agreed that it really seems like the quality and the content of all of these presentations has just gotten better and better and better each year. So I'm very, very impressed by what I've seen today, and I'm sure you're all, filled with a whirlwind of different topics here. It's pretty amazing the diverse and incredible research that students get up to here in the state of Wisconsin. And I'll remind all the winners too that so I'm going to list out the order of prizes here, but remember that there were a lot of people that competed, and you guys are the finalists. You're the winners. That's why we invited you here to talk. So congratulations is already due. Okay, so for our undergrad group, I'll have everybody kind of come up. I'll have a certificate for you, and then I'll just have you kind of stand to the side there, and we'll get all all 6 of the undergrad folks up here, and then we'll give them a big round of applause, okay? So in 5th place, we have Foster Boom. Fourth place is Ritisha Day and Domonique Olivia Valle. Third place, Jadyn Hartwig. Second place, Shaun Harrigton. We didn't work out any handshake stuff, so it's not their fault, it's mine. And first place, Matthew Dubin. Official photographer here. All right. You can be seated. Thanks so much. Okay, now for the graduate students, same deal. In 5th place, we have Kendra Wyant. Fourth place, Mindy Kim. Third place, Emmanuella, Dwomo, Agyei. Oh boy. Second place, Grant Nickles. That means that the first place goes to Claire Radtke. Just hold still for just a minute. Yep, there you go.
Big scoot this way because you're in the projector. There you go. Perfect.
Awesome. 1, 2. All right, well, thank you to all the competitors, and thank you to all their mentors and all the laboratory teams that have helped them get where they are. I really appreciate all the hard work that everyone did this year. It was a great showing, and I'm looking forward to next year. So thank you all very much. One more round of applause for all of the competitors. I believe we have lunch after this, so you can make your way down to the main keynote area to have some lunch.